Hello, welcome everyone to Deeper Clinical Results, Novel Delivery Systems for Protein and Peptide-Based Actives. I'm thrilled to have you join us today for this deep dive into one of the most exciting areas of skincare science. Just a couple of housekeeping details and the agenda for today. We'll be together for an hour, and we're going to have presentations from myself, and two wonderful experts, followed up by a live Q&A. So as I've already mentioned, on your right-hand side, you'll see several tabs. You can submit your questions anytime using the Q&A panel. And so the Q&A is specifically for questions for our panelists. And you can also vote questions, which is really cool. So we'll get to those a little bit later. I also want to point out that we'll have a little interactive polls throughout, and that's in that little interactions tab. So we'll let you know when it's time to do that, and we can have a little live interaction. All right, so I want to today our webinar is hosted by Covalo. It is the world's largest ingredients platform with over 80,000 personal care ingredients listed. It is free, and it's a really user-friendly platform that helps brands and formulators speed up product development by discovering really the right ingredients and connecting you with the right suppliers for you. So this is also hosted by Core Biogenesis. This is a French biotechnology company I'm a fan of. These have their high-value biocompounds in its unique biotechnology platform for cosmetics and the life sciences. These are the speakers today. This is all of us will be having a conversation together and also with you. Today we're diving deep into the science and the technology behind peptides and growth factors, you know, what makes them so powerful, the challenges they pose, the novel delivery system, and that is unlocking their full potential. So our goal is to leave you with actionable insights on how these actives are transforming skincare, how they can help you in your formulations and clinical studies. So I'm Alison Cutlan. I am a biologist and biotech innovator dedicated to leveraging biology, design, and the microverse to transform wellness and planetary health through sustainable innovation. Today, I will kick us off with a deep dive into peptides and proteins, what they are, how they work, and the challenges and opportunities they bring to skincare. Joining me are two incredible experts, Ameann DeJohn. She is a licensed esthetician, innovator, and product development expert who's spent years on the front lines of clinical validation. She'll guide us through how to bridge the gap between ingredient claims and measurable, meaningful results. Happy to have you, Ameann.
So excited to be here. Beyond thrilled. Thank you, Ellie.
And we also have Tony Abboud, who is the Chief Commercial Officer at Core Biogenesis. He will be diving into the groundbreaking delivery system they're pioneering to stabilize and deliver growth factors, among other biological molecules and proteins. Hi, Tony. Happy to have you here too.
Hey, Ellie. Glad to be here as well. Excited to get going.
All right, so let's do that. I'm going to start off by setting the table here and just talking about proteins and peptides in skincare. And we'll start at the beginning, the building blocks of life. So proteins and peptides are at the heart of the body's natural repair and regeneration process, and the skin's natural regeneration and repair process. Peptides are short chains of amino acids, about 2 to 10 numbers in length, and these act as messengers, really. So these signal cells to produce collagen, repair damage. Proteins, on the other hand, are more like builders or directors. These are much larger molecules, like 50 amino acids. They make up our structural components in our skin, like collagen and elastin, but they're also regulatory. So growth factors are like these proteins. They guide skin repair and renewal and differentiation. So these are really important because they support natural and normal skin functioning, firmness, elasticity, hydration, and repair. So these naturally exist in the skin as we age. But as we know, they are the buzziest ingredients right now, and with good reason. So they are highly efficacious, they're precise, they are really multifunctional. And the best part is that they're biologically compatible. So what that means is they're extremely safe. Anyone could use them, and they work extremely well in our systems because it is us. They're also sustainable, scalable, and customizable, especially when you start to use biotech. And just on the right-hand side, you can see this peptide skincare market has really boomed. It's around 2.5 billion right now, and it will increase to close to 10% every year. So I predict these are going to be in, all advanced formulas moving forward. And as the technology gets better and better, the clinicals will be better and better as well. So let's just look at and familiarize ourselves with the peptides and proteins in the skin naturally. So in young skin, there is an abundance of peptides and proteins that message and ensure active collagen production for firm, elastic, and resilient skin. But as we age, proteins and peptides decline in production. So that leads to slower collagen synthesis and slower repair processes, which of course we see then wrinkles, dryness, and laxity. And this sets up the why for skincare, right? So the goal is to help replenish what the skin naturally loses. And in skincare, there are different types of peptides and proteins. We can see that the main sort of category of these are really the signal peptides. And these boost collagen production. Peptides like enzyme inhibitors prevent collagen breakdown. Some are neurotransmitter mediated, which relax wrinkles. And then there are growth factors, which really drive repair and regeneration. And one key consideration is where these peptides and proteins work. So on the right-hand side, you can kind of see where some of these work. And, certain peptides like the signal peptides can be on the surface and they'll transmit and message the cells deeper. But certain proteins like the growth factors, which are quite large, act a little further down in the skin and really require advanced delivery systems to do their best work. And this brings us to the challenges with and opportunities really with working with peptides and proteins, and penetration is really one of them. Partly that we have worked with some of the peptides have been linked with a palmitate to help with that penetration lower, but then there's also new inventions, the Oleosome fused technology from Core, but there's also liposomes, things like that facilitate deeper penetration. There's also stability concerns, so temperature, pH, enzymatic degradation, and it's, growth factors like EGF can degrade within hours, if not stabilized. So that encapsulation or that stabilization is really important. Also, the cost and scalability has been a limitation with some of these, especially the larger growth factors and things like that. But again, biotech is really handling that in a beautiful way, especially some of the growth factors from Core Biogenesis that makes them much more accessible. So really wrapping this up, advancements in the future of proteins and peptides, we really see that bioengineering and biotechnology is leading the way here because of the delivery system, the sustainability, and also the bioidentical nature of working with some of these larger molecules like proteins, growth factors. They all have very specific folding patterns that need to be exactly bioidentical for them to work, and biotechnology can allow this to happen easily. Along the way I think we can also see personalization and adaptive peptides being in the future. I'm really excited about this. I think that biotechnology can enable some of these personalization, and also AI-enabled technologies we can look to some of this in the future. And really, I think that, they're buzzy now, but I think they will continue to be. So these are pillars of effective skincare, right? They're moving beyond that buzz to really be the pillars of scientifically driven skincare, and they're going to be more accessible. And one thing I didn't mention, there are many different types, they work in different ways, and they're very synergistic. So I can see that utilizing, many of them to target merely a global biological processing in the skin can really give incredible results. So here we come to our first poll. And before we get to the next section, I'd love to know your thoughts. Which aspects of peptides and growth factors do you find most important when choosing them for your skincare products? So is it targeted action, clinical results and efficacy, sustainable and bioidentical ingredients, or is it innovative delivery systems? So all of these might be true, but we want you to pick your top choice. Go to the interactions tab and make your selection. And we'll just kind of let these stream in. Obviously, there's no right or wrong. What we're doing is learning from you. Wonderful. All right. Excellent. It looks like clinical results and efficacy is leading. Targeted action, absolutely. All right, I'm going to let that go a little bit longer. All right, it looks like the results are in. Fantastic. And we see that 59% of those of you chose clinical results and efficacy. Absolutely. And this ties perfectly in with Ameann's presentation, where she'll explore how you can get the most out of your claims and clinical validations. Over to you, Ameann.
Thank you. I think everyone was super nice to choose clinical claims. I think they were just being really nice, Ali. Super excited to present this. After years of experience in product development, licensed esthetician, really understanding skin, I put this simple diagram together, not as detailed into the parts of the skin, because, seeing skin as an esthetician and seeing the change that would happen, I would see the surface, it would get smooth. I would see that it would get more plump. I would see that it would be more hydrated, but I really didn't know what that meant until I started doing product development. And then I would get the technical data sheets and I would see all of these wonderful claims, like increases collagen and works in elastin and inhibits MMP. And I was so excited to put all of those claims on my packaging. And then of course, learning that I wasn't able to do that. And that was frustrating. And then throughout that time of 15 years in product development, learning that you can talk about those things in a different way. And so cosmetic claims, as we all know, are really function right in this top layer of the skin. Otherwise, it's just a fluff claim. If you don't do any clinical testing, you end up with only a fluff claim. May help do something, may help hydrate, may help firm, may help your skin to be more radiant. If you'd like a clinically proven claim, you have to do clinical testing, some sort of bioinstrumentation, some sort of device like a corneometer for hydration, a cutometer for firmness. You could also do expert grading, where an expert is trained in different methodologies to grade wrinkles, dark spots, overall hyperpigmentation, smoothness, tactile firmness. You could also do a subjective questionnaire. But again, all of these here listed are really for the surface, which is sometimes frustrating when we know and we're testing for ingredients that have change underneath the skin. So I'm going to walk through the process of how we test, why we test, and what to look for. The first part we'll talk about is age versus visible age. In this slide, you see a variety of people in ages in 30s and 40s. And I chose this age group because that's often the primary demographic. In a clinical test, and when you're developing products, you first must understand who is your primary demographic. That's super important because as you're developing products, everyone will often say, well, I'm going to develop products for Gen Z because that's who's buying products. That's great, except they probably don't have the most damaged skin. You're not going to see the most change. So if your demographic is 25 to 45, you'll probably end up testing 35 to 45. And so that's kind of this demographic here. So in this particular case, you have somebody 31 years of age, and they have no lines, no wrinkles, no dark spots, and no reason to improve firmness. So they couldn't even be included in a study. And as we talk about studies, we want to be completely inclusive. And so when you're looking at higher Fitzpatrick's 4, 5, and 6, you're going to have to go up in age range to be able to see some of those problems in the skin to be able to make those changes. So on the far right, you have somebody that's 39. They still have no lines and wrinkles, but they do have some dark spots and some firmness. So if you'd like to make the claim of lines and wrinkles, you're going to have to go a little bit higher in age range to be able to make those claims for a darker or for a Fitzpatrick 4, 5, and 6. And in the middle, you have ages 31 and 41, and you're starting to see more lines, dark spots, firmness. And then in age 41, you're seeing the lines, wrinkles, dark spots, and firmness. So typically in a clinical study, you want to include 2 or higher to be able to see the changes in the skin. One of the things that we think about when we look at clinical studies as well is that why skin ages. Everyone, if you've ever look at somebody that's the same age as you, maybe that you grew up with, and they look different than you do, because everyone ages differently. I age differently. I grew up in the Midwest. I didn't have as much sun exposure. I'm also an esthetician, so I did a lot of different things to my skin. I did the ionotoresis or the galvanic, and I pushed products into my skin to work on the collagen and elastin. So I did a lot of those things that worked underneath the skin. And even though I can't claim it, I certainly did all of those things. And so smoking, obviously, we know works very much to the detriment of the skin, heat, UV damage, nutrition, all of those things. But even if you did all of those things, your skin's still going to show age differently than someone else who did all of those things. So you can't predict how your skin is going to age or how it's going to show up on your skin. And that's one of the things that's frustrating, is that you don't know exactly how it's going to age. And so some of the signs in lighter skin in Fitzpatrick 1 through 3 are loss of elasticity, redness that's easy to see, dryness, uneven skin tone. Again, and we think about those things, those are typically all on the surface. And that's when we're looking for clinical claims. But a lot of those things start deeper and they're underneath the skin. So although you're not being able to make those claims specifically under the skin, over time, these ingredients, they help to work on the surface of the skin as well. But how do you get to make those claims? Just to go back a little bit, in 1 month, this person in their 60s was able to see a change with fine lines and wrinkles with a product. That's a pretty dramatic change. In 1 month, this person was able to see fine lines around the eyes change. This person, again, we're looking at all things that you can see visually and that can also be measured with instrumentation. This person was able to see change in global pigmentation on their skin in 1 month. This is a pretty dramatic result. And this product did have some peptides in it. Don't know exact all the ingredients and everything in this one, but this was actually for dark spots. As you can see here, it's more on the surface, right? Over time, you're going to get better results, 4 weeks, 8 weeks, 12 weeks, as you start working underneath the skin, and even though you're not making claims, you're going to start to see change. But how are we going to make those claims? Again, dark spots or post-inflammatory hyperpigmentation, you're seeing change in 1 month. So a lot of times the change starts on the surface, and over time you're seeing change. So sometimes telling the story of how the change happens is helpful. So these are the areas that we look for change. Forehead lines, crow's feet, nasolabial folds, again, a little bit deeper area, age spots, under eye bags, large pores. But a lot of times you think about a claim and you say, well, I want to make a product that does all of these things. That's great. But let's find a person that has all of these things. And that's really hard. So being a little bit more targeted in who you're developing for will get you better results in your clinicals. I'm not saying your product can't do all of these things, but it's really hard to find a person who has all of those problems and still is the younger demographic. So it's not that your product can't do those things. It's just hard to be able to prove all of those things in photos. And these are the types of claims that you could get for products. You can look at after 4 weeks, you could have improvement in fine lines. You could after 2 weeks. And again, what you're looking for is successive claims, 2 weeks, 4 weeks, 8 weeks, 12 weeks. And again, you can design a study to do different things at different time points to show succession. And why is this important? So if you start with 2D, a lot of this is on the surface, but you can prove that things are happening underneath. And how can you do that? One, you can do it in succession, 2, 4, 6, 8, 12 weeks, depending on what your time points you want to do. You don't need to do them all. And you can do 3D imaging. You can do ultrasound. There's also thermal imaging to show the changes that are happening under the skin. A lot of the ingredient suppliers, they have biomarkers, and I know Tony's going to go through a lot of those different things to show that it's happening. So there is a correlation between clinical studies and ingredient technical sheets. You just have to know how to tell the story. So we have a little poll too. Allie, you're going to start my poll, right? So which area of the skin shows the signs of aging first? So is it your pores, like the size of your pores? I've ever people often ask me, do I have big pores? Do they look like big donuts? Do I have pigmentation? Do you see uneven skin tone? Do you see any fine lines and wrinkles? Am I starting to sag? How is my skin? Is it bouncing back? I'm not sure. Is my skin aging? So which area of the skin do you think shows the sign of aging first? Let's see how those interactions are coming in, Allie.
Have my microphone off. Okay, everybody is active in the interactions, which is great. It looks like, yeah, it looks like fine lines and wrinkles. C is in the lead, followed by pigmentation. Yes, because as we know, sagginess can be that sort of advanced. I'll let people finish up with their voting. Ameann, it's so fascinating to, it is interesting to align your claims with what happens in reality when you do a clinical test, right? And making sure that you set yourself up for success when you're using peptides. All right. Okay, I think everybody has made their selections. 48% lines and wrinkles, followed by 30% pigmentation. Very interesting. I would agree. Yeah. One thing I'm going to point you guys up as we go through this, and before we go to Tony, if you've got any questions at all, I encourage you to go to the Q&A section and type in anything, because at the end of this, we'll be going deeper into your questions, anything, if it has to do with clinical results or anything else. So please, you can write those down and as they come to you. And thank you, Ameann. Now we're going to go to Tony, who will show us how their cutting-edge delivery system from Core Biogenesis, this oleosome-bound technology, as far as I know, is enabling peptides and growth factors to perform at their absolute peak. So this is really groundbreaking and really fascinating talk, a really fascinating technology. Tony, the floor is yours.
Thank you, Alison. A pleasure to be here and nice to see everybody. Our numbers are really creeping up now, so this is great. So whereas Alison spoke about sort of the promise of peptide and protein actives and sort of all the different mechanisms that they act in the skin and the different layers, Ameann spoke about the need to really deliver amazing clinicals that had, bioinstrumentation and really selecting your population correctly so that you can measure changes in their skin. What I'm going to do is talk about a new technology that I think enables the next generation of protein and peptides that actually deliver deeper into the skin and hopefully give faster and more efficacious results in next generation anti-aging and skincare products. And the way I'm going to do this is talking about something called the oleosome carrier and how we use plants in order to make these wonderful products. So again, thank you very much for everybody for having me today. So whereas just kind of a little bit of a history lesson, kind of going back, a lot of the active ingredients that we use today in personal care are small molecule based. So things like retinol, niacinamide, vitamins, those are fairly small in size, they're synthetically made, and they penetrate quite easily. You have to look at irritation, but generally they can be safe and well tolerated, but they're very popular, right? They're used in almost every product in the market today. Short peptides are this up and coming category, and this is what Alison talked about, growing at 10% a year. These are 1, 2, 3, 4 amino acid type peptides. Things like copper peptides, that act on certain Botox-like peptides and things like that. They are generally synthetically made as well. They penetrate fairly easily and are growing. I think the category that's not yet really been explored and is still sort of in its infancy is using polypeptides or proteins in skincare and anti-aging. So these are proteins like growth factors, superoxide dismutase, etc. They are generally produced by the body. Having said that, though, they are unstable. They're somewhat more difficult to apply topically, and they're large, which makes them difficult to penetrate in the skin. So I think those are a few reasons maybe why the industry hasn't really progressed in that class of active ingredients yet. But let me cover a few other proteins that the body naturally makes and that may be of interest to apply topically in skincare. So we have things like with proteins like albumin, we have proteins like collagen and elastin. Obviously, we always talk about upregulating collagen in the skin. Collagen's a protein, a very prevalent protein. Lactoferrin, heat shock proteins that are involved in inflammation, superoxide dismutase that takes care of our ROS, our reactive oxygen species, and aquaporin that helps shuttle water in cells. There's also a very interesting class called growth factors. These are proteins that have a lot of different functions, but really strengthen the skin, enable wound healing, and really mediate rejuvenation. So proteins like epidermal growth factor, TGF-beta, transforming growth factor beta, fibroblast growth factor 2, FGF2. And if you look at the literature and the research, these proteins actually stop being produced as we get older. So they reduce in the amount of protein being made by the body. And that's actually one of the root causes of aging. The fact that our bodies are not making as much growth factor when we're 50 as when we were 10 or 9 or 15. So I often get a lot of people saying, exosomes are the next great thing. Bakuchiol is the next great thing. And you do get a little bit of chatter about growth factors in the industry, but I wouldn't say it's maybe as prevalent yet. So I wanted to do this little, analysis, and I went into Google Trends, and I wanted to look at what the US population is searching for on Google right now. I looked back in the last 6 months, and I was actually surprised to see that growth factors are even more popular and more being searched than bakuchiol and exosomes. So you can see here that blue graph, it's definitely an up and down sort of mountain type searching, but it's definitely at a much higher rate. There's a lot of more interest than something like the exosomes and the bakuchiol. So the consumer is primed and they're interested in this class of proteins in our industry. So having said that, the consumer is very interested, but there are still some limitations of this protein class in skincare. Number one is these proteins are highly unstable, as I alluded to previously. So epidermal growth factor degrades by 50% in less than 7 hours at room temperature. So let's pause for a second. That's not really a conducive protein to be used in skincare if it degrades by 50% in hours, right? We need to have accelerated aging and amazing long-term month stability and year stability in the shelf. FGF2 becomes biologically inactive in 6 hours. So that's some difficulties of using these large proteins. Second of all, they don't penetrate deeply into the skin. They are large. So it's hard to get past the stratum corneum and into the epidermis and dermis with the protein alone. So really, when Core Biogenesis became involved in the cosmetic industry, we wanted to see how we could apply our technology to solve some of these problems in the cosmetic industry. So namely, the delivery of large proteins or large peptides and the stability of proteins and peptides in formulation. So if we take a step back, small peptides like the copper peptides and others that are 3, 4, 5 amino acids can be chemically synthesized. So that's not a problem. That's a very well-known manufacturing method that's been scaled to large quantities. But you really can't make a 10, 15, 20 amino acid protein or peptide through synthesis. It gets very expensive. So you have to look at either isolating from human stem cells or using biotechnology to produce larger peptides and proteins. So stem cells is one approach, but not all markets allow for human stem cell derived proteins. So that's not necessarily globally compliant. Fermentation is a great approach for proteins and peptides. It is globally compliant. It's very well known. However, there's nothing inherent within fermentation that can fix the stability and delivery issues of proteins. So what we bring to the table at Core Biogenesis is an exciting platform where we use whole plants as biofactories. And really, we make proteins within the seeds of plants, and we can use the structures from Mother Nature to improve stability and delivery. And let me tell you how we do that. So we take advantage of a natural Mother Nature structure called Oleosomes. Oleosomes are how plants preserve and protect oil for the germination process. So if you think of a seed of a plant, any oil seed plant, sunflower, safflower, camelina, etc., have oleosomes. We use Camelina sativa as our crop for our plant technology. So what we do with our process is we can anchor or fuse any protein of interest on the surface of the oleosome. So you can see here depicted this green sort of image. So the oleosome is a monolayer of phospholipids surrounding a core of oil and natural vitamin E. And then anchored on the surface, fused to the surface, are these proteins. So in this case, for sake of the argument going forward, it's a growth factor like epidermal growth factor or fibroblast growth factor 2. And let's compare directly this oleosome protein fusion to just a naked protein, which we would generate using precision fermentation. Okay, so let me take you through the differences in stability and delivery of naked proteins or oleosome and protein fusions. Okay, so I covered this a little bit in a webinar with Covalo in October, so I'll go over it very quickly. When looking at stability, we found that the oleosome FGF2 fusion was far more stable than the protein alone. So this is after 12 weeks in the oven at 42 degrees. And we found that we preserved 97% of the protein after the 12-week study with the oleosome. And we only had 13% of the protein left in its naked form. So this is like a 10x improvement in stability when we fuse proteins of interest to the oleosome technology. So that's really exciting. That means we open up the doors to more accelerated stability, better efficacy for the customer because there's more protein in the product at the end of the day. This is what I want to spend a little bit more time on today because this is the new content that we're just actually presenting here today in this webinar. So it's a world first. What we looked at next was, are we able to use this oleosome carrier as a way to deliver the protein deeper into the skin. So that was the hypothesis. We believe that we can, but let's prove that that happens. In order to do that, we had to use the Epiderm FT model, which is a in vitro skin explant model with a stratum corneum, an epidermis, and a dermis layer. We apply the samples to the top of the stratum corneum, and then we look for where the protein locates by isolating each of the layers and then doing different analytical techniques. So the first study looks at GFP, which is a fluorescent protein, and where does it sort of get into the layers. And then the next study was a head-to-head of Oleosome FGF2, again, the fusion, versus the naked FGF2 protein. So just to summarize the results, the Oleosome GFP protein, we actually found the fluorescence all the way down into the dermis layer and the epidermis layer. So this was a good first signal that the Oleosome does help the GFP protein penetrate deeper into the skin. This is all measured through a linear graph, and you can actually see quantitatively the amount of protein measured in each layer. So this was a good first result that the GFP protein came into the dermis layer. Remember, the protein is quite large. It's 27 kilodalton in size. So let's talk more about the growth factor, the FGF2, which Alison mentioned. You do want to get growth factors into the dermis layer because they do have efficacy when they reach that layer. So in essence, we found that the FGF2 naked and the Oleosome FGF2 both went into the epidermis layer. So they were both detected in a Western blot analysis. You can see them here, the little band where the FGF2 sits, around 17 kilodalton. And then the Oleosome FGF2 is about 35 kilodalton. And you can see it here, the presence of the band. But more importantly, when we looked at the dermis layer, which was below the epidermis layer, we only found the presence of the Oleosome FGF2. So that would indicate that Oleosome helps shuttle the protein deeper into the skin, which is a novel sort of improvement of using proteins in topical skincare without the need for microneedling or any sort of manipulation of the stratum corneum. So I've kind of showed you that the proteins are more stable when fused to the Oleosome. I've showed you that they deliver to the dermis, but I haven't showed you that they work. I haven't showed you that they're active. So let's bring this home by sort of using some of what Ameann taught us, which was making sure you find the right people, the right population that have visible signs of aging, making sure that you use bioinstrumentation, quantitative measurements, trained professionals, and making sure that you use placebo control as well. I think that's also very important. So when we designed our study, this was a 60-person study done with a third-party independent lab. And we had 20 people using a placebo cream, 20 people using a cream with 1% the Oleosome EGF fusion, and 20 people using 1% of the Oleosome FGF2 active. That was a 28-day study, and people were using it twice a day, morning and night. So I'm going to talk about the top line of what we found in this study. So with the Oleosome EGF, this is epidermal growth factor, probably the most well-known growth factor protein. We found that we reduced the appearance of fine lines and wrinkles and improved firmness. And this was statistically significant, better than the placebo. So the wrinkle result, you can see here a pretty dramatic improvement in crow's feet after only 14 days. And this was also statistically significant. And then the firming result was just extraordinary. So we found a 21% improvement in firmness after only 14 days as measured by a cutometer. So that's pretty dramatic response after a very short period of time of using the 1% Oleosome EGF cream. The Oleosome FGF2 active, again, that's fibroblast growth factor 2. This growth factor has slightly different mechanisms. It acts really on skin tone. And we found in this case that we reduced melanin in areas of hyperpigmentation in only 14 days. This was measured by what's called a mexometer, again, a bioinstrument that measures the melanin in the area of a spot. We also found that it reduced transepidermal water loss, TEWL, which is a measure of your skin barrier. So again, very quick results, all statistically significant versus the placebo. So that's my presentation, and thank you all for listening. We still have a lot of people on the line, which is exciting, Ali. I think that's we held everybody's interest for the whole time.
It's so fascinating. We have some questions, luckily, that
we
we're
have
going
our last poll. Yeah, well, we have our last poll. So let's see if we can get through our last poll, and then we will answer our wonderful questions. So our last poll really refers to what class of proteins. Now that I've hopefully showed you that there's other actives that are coming in the market that are exciting, large peptides and proteins. What are you most excited about exploring in next generation skincare formulas? So A, growth factors, B, enzymes like superoxide dismutase, C, collagen and collagen fragments, D, keratin and keratin fragments, or E, elastin and elastin fragments. And I'll let Alison take us home with the results.
Yeah. Well, this was I'll let people think about this and weigh in a little bit. But I mean, you know, Tony, after you show such amazing results with those growth factors, I mean, the growth factors are pulling out in the front here, and I'd have to agree. I mean, increasing firmness in 15 days, that shows really deep structural regeneration, right? Yeah. So normally firmness seems like something that you've got to test after 4 weeks, 8 weeks, something like that. So it's really remarkable what the technology enables. Okay, I think everybody has weighed in. Here we go. The results are in growth factors with 74% as being the greatest interest, but then collagen fragments is also interesting to people, as well as enzymes. So food for thought for more development for core biogenesis. I can't wait to see what you guys do next. All right. So, okay, well, today, you know, we've explored the science and challenges, opportunities for peptides and growth factors in skincare. And, you know, we're really, by combining the advanced formulations, clinical results, and biotech-enabled technologies, you know, we're setting a new standard for performance. So I loved hearing from everyone, and now I'll encourage you guys, if you've had any burning questions that have come up as you've been watching, please go to the Q&A, and we will start to take some of your questions, and some great ones have started to come in. So,
Ali, I think a couple of them came in because I forgot to answer the question regarding the poll. There was like 2 or 3 that came in from that. So while the other ones are coming in, maybe I can kind of group those together. So the answer to that question is a little bit complex, because everyone's going to show aging differently, and the reason is that there's many different pathways that cause aging, right? So if you are dehydrated and you're constantly dehydrated, and you don't use moisturizer, and you don't drink water, and you live in a very dry environment, you're going to see fine lines and wrinkles, right? But if you are young and you've had excessive UV damage, but when you get a little bit older, you have been using moisturizer, you might not see fine lines, right? You might actually see pigmentation because you've damaged your skin underneath. So you might see dark spots. Let's say you're a darker Fitzpatrick, 4, 5, and 6. That was one of the questions. Would you see aging differently? Yes. So a lot of times Fitzpatrick 4, 5, and 6 will not wear sunscreen because there's been this misconception that they didn't need it. And so they'll actually have more skin laxity. Not fine lines and wrinkles, but they'll have more laxity. But then they'll have darken get post-inflammatory hyperpigmentation from dark marks more easily. So there is no one answer because everyone ages differently based on what factors they were exposed to when they age or how they age. So there's no way to really say that one thing shows up more than the other. Historically, clinical studies have been done on Fitzpatrick 1 through 3. And so it was always fine lines and wrinkles and dark spots. So that sort of answers the question very complexly. But just to kind of tie into what Tony was saying and what Ali was saying, if you start using ingredients like growth factors in all of your formulas, literally all of your formulas, then over time, you're working on the surface and underneath. And so the people are going to start to see changes continually, and then they'll just keep using the products. So.
Yeah, I think that there's no right answer there. They're all, the skin is such a dynamic system. And I think as if there are probably many product developers on the call, designing products that can work synergistically on different layers and can give really great results that are global, that different people with different issues will see, right? I love that. Okay, so why don't we go? Helena has a great question. Tony, why did you choose that plant, Camilla, to obtain the oleosome? Are there any specifics behind the choice? Was it easier to retrieve? Tell us a little bit more about that.
Good question. Thank you, Helena, and thank you, Ali. So we did use the plant called Camelina sativa. That's the scientific name. And its more common name is gold of pleasure, which is kind of a cool name, I guess, in our industry, right? You can build a nice story around that. So basically, the reason we picked it is it's very high in oleosome content. So it has one of the highest concentration of oleosomes for a seed crop. It also propagates and grows very quickly, has a very quick half-life. It grows with very little inputs, literally just water and sunlight. And it's very amenable in our greenhouse systems. So we actually grow these plants in our closed greenhouse systems in Strasbourg, France, which is right on the border of Germany, beautiful little town. And so, yeah, there's a lot of reasons why we use Camelina. We could use other crops, I guess you could say, like we have the ability to use other crops, but we really chose the Camelina as our lead crop. It also happens to have a beautiful oil profile. I mean, it's got a nice omega-3 quality to it. So not only are you getting these biomimetic growth factors or proteins on the surface, you're also delivering some beautiful oil to the skin in a very protected way. Because again, the oleosome is how Mother Nature preserves her oil. So when you buy Camelina oil for your skincare, they break oleosomes to release the oil, and that's what goes in your skincare product. So we have a way to actually preserve the intact structure of that oleosome, and it gives us all these amazing properties for our system. So I hope that answers the question.
Really cool. And Irina has asked, along with this oleosome, are oleosomes considered to be nanoparticles?
Another great question. So oleosomes are not nanoparticles. Oleosomes range between 3 and 4 microns in size. So, yeah, that's what makes it so interesting that they're helping deliver the protein to the skin. It's because the phospholipids from the oleosome are actually complexing with the lipids in your skin, and that's what's helping shuttle the protein into the skin layer. So it's not nanotechnology. It's a micron technology.
And I have a question about this. So when we think a lot about encapsulation, right? It's like, let's just say a growth factor, it's encapsulated by a liposome. It's on the inside. What's so interesting about this technology is that the growth factors are on the outside, and you don't just have one. It looks like you have several all the way around this droplet. So does that mean better efficacy? Not only that it goes in, but you've got a lot more of them that you're able to attach.
Yeah, great question. So I think like liposomes and other things will typically encapsulate small molecules, small molecular weight active ingredients. Peptides and polypeptides and proteins are very large. And they have to be folded, and they have to have structure to interact properly with the receptors in the cells. So we use the oleosome as a carrier, but then the protein is very close to the surface, but it's properly folded and exterior so that it can interact with those receptors. So, and it still delivers it. It's, as I showed you, it stabilizes said protein, but it retains its structure, I think is what's important here.
That's really cool. What is since we're on the oleosomes, can you give us an example of the inky for this type of?
So the inkies are all, so we have two, we didn't really talk about this per se, but we have two ingredients that are available to the personal care market globally. So there's Povita and Poforia. Povita is EGF oleosome, and then Poforia is FGF2 oleosome. Those inkies have already been approved by PCPC, and it's a little bit long, so I don't know it exactly, but it's camelina sativa oleosome brackets with your SH polypeptide 1 or SH oligopeptide 1, depending on what growth factor you're talking about. And the ingredient is in a blend of xanthan gum, water, glycerin, and gluconadelta-lactone. So, because if you know anything about growth factors, you need like very, very, very small doses. So when you use 1% of Povita, you're actually talking about 0.0002% protein. So we had to disperse it in a way that made sense and that was easy for you to work with as a chemist and a formulator. So that's the inky. I'd be happy to send it to you afterwards.
And along that, but formulating with this how's the integrity of these?
So great question. So you can formulate with them. Typically, we add them after we cool down the batch a little bit below 45 Celsius. This is a protein, right? A protein ingredient. So you have to be kind of mindful of that. So our customers will create their oil and water emulsion at temperature. They'll start to cool it down, and then they'll add the active at the end at 1%. You can mix them. It's very pH tolerant. It's tolerant with a lot of other ingredients. We, in fact, have some prototypes as well you can use as a base formula, but it is pretty forgiving in a formulation.
Any ingredients that don't work well with growth factors?
You know, anything that would be disruptive or denaturing proteins, so enzymes, but we have found that the vast majority of ingredients that you would use, common ingredients, are very tolerant. So, you know, maybe that's a question we can take offline. If they have a specific formula and they have specific ingredients, we can look at that, have our team look at that. But generally, it's a forgiving technology.
And pH?
4 to 8. pH between 4 and 8.
Oh, that's pretty forgiving.
Yeah, exactly.
Yeah, that's pretty forgiving. So here's a question from Elena. In terms of the safety of growth factors, and is there any undesired growth that could be of concern when using them?
So that's a good question. So like we are not medical doctors, or you know, we're simply making a class of proteins better, right? That has been used for over 30 years in skincare. So in the 1990s, a plastic surgeon by the name of Dr. Brown was experimenting with EGF on wounds and found, you know, amazing dramatic wound healing. And ever since then, there's been thousands of research and publications, and dermatologists and researchers have all looked at growth factors in skincare. There's over 150 brands that are using growth factors today in skincare. So it is a class that's been well described, well, you know, looked at. We conduct all of our extensive safety testing, and that's large HRIPT panels, that's irritation studies. So we feel very comfortable with the technology. It's got a track record of history and of safety and performance. So I would really consult the literature to really dig into it for your own needs, but we are pretty confident with it as a category for sure. I mean, our body naturally produces these proteins. And all we're doing is introducing topically miniature doses to complement that on your skin. So I think if that's helpful in any way, those are some comments on that.
And I think we've got time for maybe one more question, and I'm going to bring this one to the top. You touched on exosomes. I think these are a really hot ingredient because these do contain growth factors in them, right? They're little delivery, natural cellular delivery vehicles in their own right. Can you briefly tell us why oleosomes, how they might compare with exosomes?
Yeah, so it's a great question. So exosomes again, I'm not an exosome expert. I'm an oleosome and protein expert. But from what I understand from exosomes is they are much smaller. These are nano vesicles. We are micron size. So these are nanovesicles that that engage in cell signaling and help cells sort of signal between each other. From what I do also understand, though, is you really have to look at the source of exosomes. So a human-derived exosome would probably be more beneficial than some other exosome because that's going to function with the human receptors. The great thing about our technology is we're using oleosomes from plants, but we're using biomimetic growth factors. So proteins that have the same sequence as the human sequence. So you get the benefits of the oleosome and that it's plant-derived and it's not nano. And then you get the benefits of these amazing growth factors that do a lot of the heavy lifting and anti-aging longevity. So we haven't run head-to-head trials against exosomes or anything, but we have this belief that we may be able to differentiate ourselves favorably against exosomes in the market. Because if you look at the exosomes, it is a lot. Growth factors are a lot of the reasons why they work. And we happen to have them in this purest, most, carefully folded form. So that's my approach. Again, if you're interested in exosomes, please go look at them, but this might be a nice complementary or replacement technology to that.
Great. All right. Well, we'll have to end there in terms of the Q&A. Great questions. If you want to learn more about Core Biogenesis, I'm going to direct you to the Cavallo website. It's free of charge. If you go and click on ingredients, I don't know if you can see but up at the top, you'll click on ingredients and you'll see their full ingredient database. Simply type in Core Biogenesis in the search bar or on the right-hand side, and you'll see the two ingredients, Povita and Porphyria, that show up. And then let's say you can click on one of them and you'll get, all of the details and the documentation about this ingredient. So you can request samples, quotes, and learn a little bit more about, how to formulate with them. So thank you so much for joining us. Thank you, Tony. Thank you, Ameann. Thank you all for being here and your thoughtful questions. We hope we're leaving you with fresh insights to bring back to your work. So shortly after this webinar, you'll receive a follow-up email with our email addresses in case you have specific questions and you want to get in touch, along with some other useful links. And I think the replay for this is going to be available later on in the week, but you will get that via email as well. All right. Thank you, everybody.
Take care.
Have a great week.
Bye-bye.